Hey guys, I'm Tony Tian, and if you're new to this channel, I'm a second medical student at University of Otago. So yesterday we talked about some of the histological features of the early part of your GI tube, of your esophagus, and also your oral cavity, and also some of the tissues at your oropharynx, etc. And basically some of the histological features, what kinds of cells is it made of, and what some of the features of these cells. Alright, so today what we're going to be talking about, so we're going to just extend from that, except these very surface protective and fundamental surrounding structure, your tongue, the gross observable structure with your eyes. We're also going to talk about some of the hidden ones. So today we're going to be focusing on the glands, the secretory glands, and because we're talking about sort of the early upper GI tube, we're going to be talking about the salivary glands. Right, so basically there are two kinds of saliva, and if you have some sort of life experience, you would know there's the really foamy one, and there's the very liquid one that contains a lot of enzyme that actually helps you to break down some of the glucose in your food. So if you have been chewing rice or bread for a really long amount of time, you might notice that you can taste something sweet, and that is actually because these enzymes, these amylase, are breaking down some of the sugar into simple forms of sugar that can be tasted with this sweet flavor. Alright, so there are three types of major cells in your glands, and this is the mucus secreting cells, the serous secreting cells, and something we call a myoepithelial cells, with a basket cell. And basically, it actually forms sort of like a basket-like structure around the outer perimeter of the acines, and basically just imagine them as hands squeezing like, imagine a cow, so they will have the mammary glands, basically just the milking tits, and you need to need some hand to squeeze this mammary tissues to force the milk out. And so same over here, you need to sort of squeeze these acids to form the secretion out into the duct. Alright, so now let's talk about the different features of all these different kinds of cells. So first, to start off with, we're going to talk about the mucus cells. So mucus cells has this cylindrical shape, and it has a flat basal nucleus, and it has a pale mucus droplets at the apex, and it secretes thicker and more viscous saliva, and through a larger lumen of the acina in comparison to the serous cells. And also the mucus, because the mucus cells are larger, the mucus duct are larger, the mucus acinus is the acinas are usually larger and more elongated as well. And then you have the serous cells. So these are usually pyramidal shape. They have a round basal nucleus with a lot of rough endoplasmic reticulum, and you have a lot of sandwiching granules at the apex, and it secretes the watery and the serous saliva through the narrow lumen of the acina. And so basically these, because they are slightly smaller, they have this round and less elongated shape, and the zymogen granules actually contains a lot of the digestive enzyme, and basically this is where all the amylase of your saliva is coming out of. Right. So now we talked about the acinus, and the secretion actually comes to the duct. And for the cells that's forming the duct, it's a slightly different type of cells from the secretory cells. And so these serous membrane will go to the initial duct, the small lobular duct that's formed of the simple low cuboidal epithelium. Low simply means they're short, and then it goes into the striated duct and the interlobular duct, and then it goes into this duct that has the stratified columnar epithelium, and that's the main duct. And basically it's generally, so just remember it's generally lined by cuboid or columnar cells, and it's never the squamous cells. And part of this reason is because the function of these duct cells actually has the function of modifying the pH. So it does, the duct cells, even it's in the duct, it's just for sort of intuitively you would think it's just for transmission, but also it also secrete substances and modulate the saliva bit by bit. All right. So basically, in these cells, the nucleus are located more towards the apex compared to the secretory cells, and it has basal striated cytoplasm. And basically, what that means is just the basal interdigitation of the cytoplasmic processes. And basically, what that does is that increases the surface area. And you have columns of mitochondria at the end, and this is because these cells are very active, and this is, as I mentioned, is because it has a lot of secretory functions. It needs to modify the saliva and regulate the pH, and from time to time it does secrete lysozymes and IgA as well. So lysozyme is basically a type of actually I'm gonna confirm this, but from what I remember is a type of substance that actually destroys the cells. Right, so it is a membrane-bound organelle with the roles and process involved in degrading and recycling cellular waste. So basically just breaks down the dead tissues, the dead cells. And then IgA is a form of antibody. Right, and another thing about the duct cells is because it has a columns of a huge amount of mitochondria, and mitochondria are very stainable under the stain, so it appears to be very dark during the histology. All right, so that's everything I'm going to be talking about today. I hope you guys have a general understanding about the glands, about the salivary glands. And if you have any more questions, just leave it in the comment section below. And if you like the video, hit the like button and subscribe to the channel. And I'll see you next time. Bye.